Hello, everyone. Good morning. Almost good afternoon. We're going to get started. I am so delighted to be back in the classroom with you. It means I have my mask back on, but it means maybe we'll get to chat for a couple minutes after the seminar today. As usual, we have an excellent guest speaker for you today, and before I introduce our guest, I have just a few announcements. Okay, so first, your final assignment is coming up. And I know that some of you are starting to have questions about it, and Sam and I will post updates to the assignment on Canvas before next class. And so once we do, we'll send out an announcement, and we'll let you know that you can go and check. So know that that information is coming. And in the meantime, please keep your thoughtful summaries and questions coming all or most of the weeks that we have left. It's been really wonderful to read how these seminars are landing with you, and as I think you know, we pass on many of your questions onto the guest speakers afterwards so they can know what they've left you with and kind of what some of your interests are. So we did do that after last week's presentation with Professor Fakhri, the UN Special Rapporteur on the right to food, and actually we have a pretty fabulous invitation to continue the discussion with him this Friday in Professor Drunowski's Global Nutrition class. So hopefully you saw that announcement that Sam posted yesterday. It has a Zoom link. Please don't share that Zoom link with anyone, but know that you are invited. Our whole seminar has been invited. There will be an opportunity for Professor Fakhri to answer some of your questions, some of the Global Nutrition classes questions, and then engage in some more of a dialogue than we had opportunity to do in the seminar. So I hope that some of you will be able to make it and continue that really valuable interaction with this insightful global position. So it was really exciting also to read how that seminar landed with you, how much you learned about the United Nations, about those particular roles, and then, of course, some of the local to global controversies that are playing out in food systems at this moment. So today, we have an opportunity to move on to a different seminar, a different topic, but before I introduce our guest, I wanted to just say, as you can see up here, the focus today will be on agricultural water, on irrigation water quality in agricultural production systems. And it may appear, a bit of a surprise to have a lecture on the right to food next to a lecture on water quality. But I want to assure you that under the framework of food systems, these are actually interconnected systems. So if you recall back to some of the food system graphics that we started the seminar with, these system of systems that are interconnected, interdependent, and of course, our overall understanding of food systems is inherently interdisciplinary. So from last week, you'll recall that Professor Fakhri was talking about access to adequate and available food, and he got into all the nuance of that. And very closely connected with those concepts is food safety. And that, of course, is reliant on so many inputs, including agricultural water quality. So with that, I am so pleased that we have an expert here to talk us through this concept, Dr. Karen Levy. Dr. Levy is a professor in the Department of Environmental and Occupational Health Sciences here in the School of Public Health. And Dr. Levy's research group is broadly interested in exploring the topic of how environmental change affects transmission and incidence of infectious diseases, with a specific focus on the ecology and epidemiology of waterborne diseases. So, Dr. Levy, thank you so much for being here. I want to invite you up, and we can all give Dr. Levy a nice, warm, in-person welcome. Thank you.
Thanks. Great. There's not a clicker, right? I'll just... Okay, great. Yeah, so if we think about the right access to food, we also want that food to be safe to eat, right? So as Dr. Sippo said, I'm going to talk about agricultural water. And I want to preface this by saying that my expertise is more on microbial water quality and not as much on chemical water quality. That's an entire other topic that is very important to the production of safe food and also to occupational health of food, people working in the food industry, but we won't really touch on that today. So what we'll talk about today is about, irrigation, water quality, and agricultural production systems. So first talk about just how is water used in fresh food production. And then we'll talk about foodborne disease risk and how we study that and how we examine that. I'll give a couple of case studies of some high-profile outbreaks of E. coli in fresh produce, and then also my own work on Salmonella in South Georgia. I used to work at Emory University in Atlanta and moved here in June 2020. So I was working in Georgia. Hopefully in the future I'll get involved with issues in Washington State. And then just talk a little bit about how do we regulate this and how do we govern these issues. So starting with the first one. First of all, what percentage of the world's water, fresh water, do you think goes for agricultural production? Raise your hand if you think it's less than half. Raise your hand if you think it's more than half. Okay, raise your hand if you think it's more than 60%, more than 70%, more than 80%. If you actually go and look globally, in terms of global consumption of water by sector, the vast majority of the fresh water that we're using across the globe is going towards agricultural production and for irrigation. So this is one thing that's really interesting about these graphs is that they separate out irrigation water from water used for livestock. So does anybody have any suggestion why that's like how do you decide which category it falls into? Yeah. Yeah, you want good quality of water to feed to the livestock, but what else is being fed to the livestock? What's that?
Plants as well as
Yeah, there's all the water that you need to actually, like the cow has to drink water, right, and has to maintain their facilities. But in addition, all of that grain or any other feed that's feeding the livestock also needs water to be produced. So right in these kinds of graphs, just keep in mind that that's in the category of irrigation. But a lot of that is actually ending up going towards providing feed that ends up in livestock. So it's a little bit misleading to have these tiny little slivers attributed to livestock in a figure like this. But anyway, the point of this is really just to say that a lot of the fresh water in the world is being used for agriculture. And here's a graph of a map of the U.S. just showing the vast amount of acreage of irrigated land in the United States. So there's a lot of different ways to irrigate agricultural fields, and you probably have seen some of these driving around. Some of you might have agricultural backgrounds and familiar with these different types of irrigation systems, ranging from surface irrigation, very advanced center pivot, programmed systems to irrigate at just the right times of the year, drip systems, which help conserve water, all the way to manual irrigation, which means like you're actually going around, physically irrigating the land. There's also, so these are the things that we kind of think about when we think about water used in agriculture is the irrigation water. And I'm not going to go through this whole list, but just so you know, there's this other kind of water that's used in agriculture. It's foliar contact water. So anything that is actually in touch with the plants there's just a ton of different ways that we're using water and applying the water to plants for all kinds of reasons, not just related to the actual watering of the plants. So when we're applying all this water, it's really important that water is the quality of that water affects the risk of foodborne disease. So foodborne illness, there is 48 million foodborne illness cases every year in the U.S., and greater than 46% of this is attributable to produce. I have the category up here. So, and this is something that's kind of risen to the surface, risen to prominence, the idea of produce related to foodborne illness, really after this big E. coli outbreak in 2006 that I'll tell you about in a little bit. But as people have started to look into it, the amount of foodborne disease attributable to produce is really a big number. And so there's a lot of people thinking about that. And when we think about foodborne outbreaks, this is from my colleague Mia Mattioli, who's at the CDC, this slide, and the definition of foodborne outbreaks is that you just have 2 or more people epidemiologically linked after exposure to a food source. So all you need is 2 individuals to count as an outbreak. Much of the time, if you just have 2 individuals, it's never going to get reported. And much of the time, it's going to be much greater. But that's the actual definition. And so you can have all kinds of different illnesses that result from that, mostly gastrointestinal illnesses. And there's a ton of different foodborne diseases. They're caused by parasites, bacteria, viruses, toxins, chemicals. So really, like, a lot of different things responsible for foodborne illness. With the top 5 are listed here. And a lot of those like norovirus in particular, is a lot of outbreaks of norovirus are related to food, and to produce. And so anyway, can keep going. So how does produce get contaminated when it's out in the field? So the first thing is it can happen out on the field. You can have during the production process, you can have water, dirty water irrigating produce, and that causing contamination of the products in the field. It can also happen during the harvest process. And a lot of the actual harvest methods can really determine the risk of if there are pathogens present on the landscape during the production system process. When you go to harvest it, how you do that really can have a bearing on how much risk you introduce into moving those pathogens from the field to the actual product. So a great example of this is the spinach. In the spinach outbreak, one thing that was noted is that there's kind of these harvesting machines that will cut the plant on the field, and they'll kind of come down and like mow the product. And so then that cut piece of spinach then can be in contact with the soil, and that produces an opportunity for transfer of the pathogen from the field to the plant. So some of these things are really important to think about, as like, what are the harvest processes? And then there's also the processing plants. So you're using water, a ton of water in processing of the product. And so lots of opportunities for, if that water gets contaminated, to contaminate the product. Also in the machines, you can get biofilms built up in the machines, and that can be a place that harbors a pathogen, and then it can propagate within the production system in that processing plant. Then you also have to get things from the processing plant distributed out to the stores. And in that process, you can have people, you can handle people having dirty hands or whatever, some different aspects of keeping the product hydrated. All of that can introduce opportunities for water to contaminate the product. And then you get it to the store. There's sprayers. When you go to the grocery store, you see those sprayers misting all the produce to keep it nice and fresh. If there's contamination there, that's an opportunity to contaminate the product. And then also when you get home, if people aren't managing their own food safety in their homes, that's another place where it could be introduced. So and each of these different pieces, of course, has different capacity for different sizes of outbreaks. If it's just in your home, you didn't wash your hands after you went to the bathroom, you made a salad, you had a dinner party, you might have a few people who get sick, but if you have a contamination event during the production or harvesting process, that is really an opportunity for a wide-scale outbreak kind of an event. So the size of the number of people that it could influence really matters. And as you can see, through all these different steps, it's very hard to trace back, well, what was the point when it got contaminated? There were so many opportunities to get food contaminated. How do you trace it back to that, where it was introduced, where that contamination event was introduced? And I'll show you a slide in a second that talks a little bit more about that. But in terms of how we determine that food was the cause of an enteric pathogen outbreak, there's a lot of different types of evidence we can use. So the first is just your kind of run-of-the-mill epidemiological outbreak investigation. So this is kind of standard epidemiological process. People go to the classic is like, there's the macaroni salad, potato salad at the fair or the party, and so you do an epidemiological traceback investigation. You ask people what they ate. The people who got sick, they all had the potato salad. You trace it back to the potato salad, right? So you have these patterns of association between illness and exposure based on who's eating what and who all had a common source. So you go through these just standard steps of the outbreak investigation so that then you can go back and control it. There's also the idea of these traceback investigations where you do your epidemiological investigation, you realize everybody had, Joe's farm cucumbers. And so you then can go back to Joe's farm and try and collect samples at that farm to look for the spot that has a lot of contamination. And so this is something that the CDC does a lot of. This is the slide I was thinking about before in terms of traceback. Also, the complicated piece is that you have different pieces coming from lots of different places, and you have mixing of different produce types, right? So I love this slide, the well-traveled salad. You can have stuff from all over the world, all of the different pieces that are contributing to your meal. And so it really makes it very difficult to trace a lot of these things. So then when you go back to Joe's farm, you want to go back and you want to, sample the environment, see where there might be a problem in operations. But you have a lot of issues to face. One is you might go back there, it's already gone. It's died off, there's been dilution. It might not even be findable at all. You don't know where to sample when you show up at Joe's farm. Do you sample, from the creek that's running through? Do you sample from the wash water? You don't have unlimited resources, so you have to decide where to sample from. You're looking for a needle in a haystack, basically. The pathogens are usually there in pretty low abundance, and so you're trying to find something that once it's in your gut, it's really easy to find in a stool sample because it's amplified and you've got a lot of it. But when you're looking for it in the environment, it's really a needle in a haystack problem. A lot of times it's really difficult to culture isolates from the environment. There's time delays between the contamination event and when you can get out there to sample. You had to have gone through that whole epi investigation. And then there's also potentially maybe multiple places where the pathogen could have been. But this is kind of how it's done. The CDC will go out and they'll either go and take a grab sample. You can take a small volume sample, 100 mils or 1 liter, and test for just general contamination. And then the other thing that they do is they use these ultrafilters, which are really kidney dialysis filters. And what they can do is they can process very large volumes of water. So you could put that in a creek. You can run a pump and filter 100 liters of water, send that back. That cartridge gets sent back to the CDC, and they can process it for a ton of different pathogens and look for what they're looking for. So that's something that's been done. It was a technique that was developed in response to some bioterrorism initiatives when anthrax came out, and then they've basically got a bunch of money to develop it for anthrax and now have repurposed it for all these different other environmental investigations. They can also sample for soil and sediment. In the case of, this wouldn't be in the agricultural sector, well, I guess it could be. You could do like a nozzle or aerator of a water tap, and various different kinds of filters. So that's often how this outbreak investigation can be done. And then the third one is that you can really, now we have much more advanced techniques that we can do these microbiologic traceback investigations where we can really look for the fingerprint of a particular pathogen and identify. So you go and do your traceback. Well, maybe there's Salmonella over here, but there's also Salmonella on, there's Salmonella on Joe's farm, there's Salmonella on Bill's farm. How do you decide which one was the origin? When you get these techniques to really get this molecular signature of the pathogen, you could potentially trace, you could figure out that actually that pathogen came from Bill's farm, not Joe's farm. So you're looking for an identical pathogen that causes illness in the food, on the farm, in the growing environment, or in the facility. And as we've gone along with this, as techniques have developed, we get more and more specific. So this is an example for Salmonella. It used to be there were serotypes. Then we got into this thing called pulse field gel electrophoresis, which was like these with bands. This is actually an image from that, where you would get these little bands based on digestion, and you could look for matches. And there was a whole system CDC developed to try to look for these matches in the bands. And now there's a transition going to whole genome sequencing, where we can actually identify individual SNPs that are different. And this is an example of where they're comparing different isolates. And so the ones on the left are all not related at all. And then the ones on the right all look really, really similar. And you see those in blue, it says it's the same PFGE, but not part of the outbreak. So you had the same, if you were just using the PFGE method, you were only able to identify, you could have thought everything was part of the same outbreak. But with whole genome sequencing, we can now get way more specific to understand that what actually counts as an outbreak. And you can see there they had cases in Maryland and New York, and they all had this matching whole genome sequence pattern. So these are the kinds of techniques that now the goal is that if you use these techniques and you can get state labs up and running so that they have the capacity to do this. Here's an example of an epidemiological curve of the number of days and number of cases of a foodborne disease outbreak. The idea is to catch it much earlier, before it goes all the way to market and before a lot more people get sick. If you could identify it, trace it back, cut off the chain of and let people know that Bill's farm was actually the source of this. Don't throw away any product from Bill's farm. Then you're going to catch it that much earlier and save a lot of illnesses. So that's kind of the big picture of foodborne disease risk. Does anybody have any questions about that? Before I jump into some case studies, yeah.
What is the repercussions when you find foodborne diseases on farms?
Yeah, that is a great question. The question was, what are the repercussions if you find a foodborne disease on a farm? And it can be very, very costly, both for individual farms and also for just whole industries. Because if you're like, well, the peanuts that came from Georgia are contaminated with salmonella, then nobody wants to buy peanuts from Georgia, even if it was isolated to one bad actor on one farm. So a lot of what happens, and we'll talk about this a little bit when we get into the regulation, a lot of what happens is that you can't actually enforce a lot of the regulation, and it's a lot of peer influence, trying to keep that from happening at any given farm so that you don't destroy a whole industry in a particular region. It's also really costly to invest in these traceback systems. So now, if you're a big enough farm, you need to be able to have these systems in place that let people know, like if you bought a bag of cucumbers from Costco, you could trace it back to Joe's farm, and Joe has to invest in the system that makes it possible to do that. And that's been something. So there's a lot of economic repercussions for farmers. So let's talk about a couple of case studies. The first is this high-profile E. coli produce outbreak. That first, it happened there was this big spinach outbreak in 2006, and it really woke people up to the idea that produce could be a concern on a large level. And there was this big food safety recall, and I was in grad school at the time in California, and this happened in California, and it was really scary. You know, you'd go for your breakfast, and you couldn't get the omelet with the spinach, and everybody was talking about the spinach at the time. So, and it was linked. One of the reasons why it was so bad is that it was linked to this kind of pathogen called E. coli 0157H7, and it causes really severe impacts on the body, and it leads to hospitalization and sometimes death. So there were, in this case, 225 cases with 39 people developing hemolytic uremic syndrome, and 5 people died. And so you can see here, it was in August. There was a contaminated lot of spinach produced, and then a bunch of cases happened, and then it wasn't until 9 days later that the outbreak strain was isolated from spinach in that person's refrigerator, and then there was a whole bunch more isolated before, and then all the spinach was pulled from the shelves, and the epidemiological curve goes down, which is typical. And this was traced back to the Salinas Valley in California, and this is a picture of the outbreak investigation, and they were able to see around this farm, they were able to find the outbreak strain, or they were able to find not the exact strain, but E. coli 0157H7 in cattle, wild pigs, water, and soil samples on these farms. And when they went and looked at a particular, the place where the contaminated lot was produced, it was a facility that also had cattle and feral pigs. And so then these pigs, the idea was, well, cattle are really the problem. They're the ones who carry 0157H7. But then you have these feral pigs that are just out on the landscape running around, so they can kind of be a vector of the pathogen and carry it to other locations. And it really caused a lot of implications for wildlife, because there are these wildlife corridors that conservationists were trying to make it so that wildlife can move in this agricultural growing region, and people were cutting down all riparian borders, keeping animals from being able to move across the landscape, which ultimately it was really the cows and growing produce so close to the cows, but all the wildlife kind of took the hit for that. So, you would think 2006, there was all this, attention paid to this big spinach outbreak, and then, Amber Alert. No worries. I'll just keep going. Then you get back to 2018, and Mia, who had provided the slides in one of the previous slides for me, she was involved in this. She was my postdoc, and then she went to go work at CDC, and then I was having coffee with her, and she said, "Oh, you're about to get an alert on your phone of a news event that it was right before Thanksgiving." And everybody said, CDC says, pull all the romaine lettuce off of your shelves. Throw away all romaine lettuce you have, because it's been tainted by E. coli 0157H7. And this was kind of, so she was involved in this environmental assessment, going out to the farms where these products had originated. And it was in the Yuma Valley in Arizona. And what they did is they go out, they do this ultrafiltration sampling of irrigation water, and found that there was an exact match between the 0157 that was in the irrigation water with the strains that caused the outbreak. And yet again, it was basically traced back to these irrigation canals. I don't know if you can see here, right in the middle, there, you can see the irrigation canal kind of running through this agricultural land. And then you have a cattle pen drainage retention. So like you basically have the cattle right there growing right next to the produce growing fields. So ultimately that's kind of, from my perspective, where the problem is, but we don't seem to learn our lesson. We're still growing produce in the same areas as we're farming cattle. Yeah. Yeah, they're definitely, so one of the questions was, do they spray the manure from CAFOs onto growing fields? And there's a lot of rules about that, about when you could do it, what the products might be. So you're certainly not supposed to do that for like spinach or lettuce or something that you're going to eat raw. It would be more for like cotton, that you could use the manure for agricultural production. But, you know, there's wind, that if you have the cotton field is right next to the lettuce field, then you have potential for cross-contamination between those fields. So there are a lot of rules to govern it, but it's also like, it's not a perfectly closed system. And ultimately, food grows in the ground, right? And the ground is not going to be sterile. We don't want it to be sterile. So this is another example of a case. This is a system that I worked on, which is Salmonella in South Georgia. And salmonellosis incidence in Georgia is higher. It's elevated compared to other regions of the country. In Georgia, it's 18 per 100,000 compared to 14.9 for the rest of the country. And actually, Florida is really high too, but they don't report their numbers, but it's even higher than Georgia. This is one particular strain of Salmonella called Salmonella javiana, and you could see it's really concentrated in the southeast. And so there's 2,500 different serotypes of Salmonella, but there's just more incidence in the southeast, and then certain strains like this, which I think of them as environmental strains because they're in particular kinds of environments. The CDC calls them geographically specific strains, I think is something like that. But because they're concentrated in certain locations, but it kind of points to that there are these environmental reservoirs and pathways of Salmonella exposure. And in this area of Georgia, there's 80,000 irrigation ponds. So each of these little ponds is a surface water pond that people build, put a little dam and create a water source, and then use that as irrigation for the farm. But unfortunately, in many cases, there's not a lot of treatment of the water in most cases before they apply it to crops. So it goes straight from the pond to irrigation systems to the product. And in our work, we found that Salmonella was detected in all of the irrigation ponds we sampled, different concentrations, different times of the year. It is really low concentration, so the risk is unclear. It's like, it's there, but would it die off by the time it gets to market? But it certainly is a possibility that it's there. And it could certainly seed a processing plant, for example, and then, kind of have its own, get a life of its own in that kind of processing plant. So we've done a lot of work in this area. We looked at storm runoff to try to understand where it was coming from. Or we like pinned these bags overnight. We're chasing storms and collected water running off of the fields. And we found that the streams feeding the ponds were 100% were positive for Salmonella. Runoff from the fields was lower, so it seems like it's more like in the water systems, and runoff from forested area was also lower. So the streams are concentrating the pathogen. And then we also did these rainfall events where we looked at the concentration before and after storms and found very elevated rates of Salmonella after the storm compared to before. So that has implications for timing of irrigation. So thinking about safety measures like maybe not having a waiting period after a storm to irrigate your field. And then we also did PFGE typing of what we were collecting on these farms, and we found matches with like between the pond and the field, and then the field and the produce. And we had 2 hits on produce, on cantaloupe and cucumber, and we had 1 PFGE match between the irrigation system. We actually like went out and like captured samples directly from the sprinklers or from other irrigation system types, and we're able to find a match. But the matches were not across all 3 sampling locations, and they weren't necessarily like in time. It wasn't like on the same day we found it in the irrigation system and on the produce. So it wasn't like a super tight connection, but they were out there and there were matches. And again, it's kind of like looking for a needle in a haystack. And then lastly, we did all these experiments where we were looking at the issue of like, okay, if it's on the field, how does it actually get onto the plants? Can it get onto the plants? So we did these splash experiments, and these were really cool. We had this rainfall simulator and set up a rainfall simulator so we could create rainfall events. And then each of these little things is like a pan where we were collecting water that splat at different heights, so you could see how far it splashed from the ground to the field. And the thing that we were interested in looking at on these experiments was what the prior conditions were. So if you, we kept an area dry, we covered it and didn't let rainfall fall in the soil, and then another area where it was just wet. And if you applied the rainfall, if you had a heavy rainfall event, after it had been dry, it was not as risky as if it had been wet, and then you can kind of splash, and you can imagine the splash kind of splashes up more. And so we did all these experiments, and this is my PhD student, Debbie Lee, and we found that Salmonella in the soil can be transferred to produce via splash water up to 80 centimeters in height, and it could survive in the soil and on produce for 8 to 10 days. And that the transfer is greater when the soil moisture antecedent to the irrigation or rainfall events was higher. So trying to come up with ideas of ways to think about, management practices that could reduce that transfer. Okay, so all this is happening, as I said, we know food grows in the ground. We know it's not sterile. We know there's these pathogens on the landscape. So how do we handle that? What do we do with it? And there's been a lot of action in the last 10 years or so trying to come up with regulatory frameworks to deal with this. And so the first thing is that the FDA has guidelines to minimize the microbial food safety hazards for fresh fruit and vegetables. And I think the really important thing to keep in mind is this is guidance. So it's not regulation. What the FDA has is not regulation. And the idea is to have prevention of microbial contamination. And the idea is to try to rely on prevention rather than corrective actions once the contamination has occurred. And also have accountability, across all these different levels of agricultural and packing environments. And so the FDA does these GAPS programs, that's the Good Agricultural Practices. And so the idea is to create these gaps and do trainings for growers so that they understand what are the best practices and best ways to minimize contamination. So they're supposed to be there to assist people, to assist the growers, to increase awareness, provide suggestions to minimize the hazards. And it covers all these different water uses, also manure application, like you shouldn't, be applying your CAFO manure to a lettuce field. Things about worker health and hygiene, sanitation, all of these different aspects. But they don't impose any new requirements or supersede existing laws or regulations. And this is kind of like the idea is, a lot of like university extension workers will go out and help growers learn about what these good agricultural practices are. There's certification programs, et cetera. And there's things like this. This is just some examples. Like for water, wherever water comes into contact with fresh produce, its quality dictates the potential for pathogen contamination. You want to identify your sources, know the use of the land, maintain wells, all these kinds of things. Aspects of processing water, cooling operations. So there's just a lot of sort of guidance about best practices. So then, while that's been going for a long time, but now there's also what's called FSMA, the Food Safety Modernization Act, and there's the Produce Safety Rule of FSMA, and that was finalized in January 2016, so a little while ago now, but then it took a long time before it was actually enforced, and it's still kind of just coming online. But it's the first time that there's actually science-based standards for safe growing, harvesting, packing, and holding of fruits and vegetables grown for human consumption. So it applies to agricultural water, biological soil amendments. There's a whole separate rule for sprouts. If you study food safety, the one thing that I can tell you, the one thing I'm just not into eating is sprouts, because sprouts are very hard to clean, use a lot of water in production, and are prone to contamination. A lot of outbreaks trace back to sprouts. Management of domesticated and wild animals, worker training, health and hygiene, equipment, tools, and buildings. So all of these are part of the produce safety rule. And so one of the big things that it's done is establish ways to monitor water quality on the farm. And there's this E. coli standard, and it establishes E. coli concentrations and monitoring frequencies for agricultural water quality. Well, E. coli is a very imperfect tool. So E. coli is an indicator organism that we use in water production systems. So City of Seattle, your water treatment center produces water. You want to make sure there's no E. coli after it's gone through the extensive water treatment before it goes to you in your tap. And if you find it there, then that's problematic. E. coli, if you go out and measure, go outside and measure the soil and measure, kind of like rainwater runoff in the next time it rains, which is every day here, you're going to find E. coli runoff. It's kind of everywhere. And so the growers were like, this doesn't make sense to apply this E. coli standard to the agricultural water system because you're just going to shut us all down. So there's been a lot of back and forth to try to figure out what is the best standard. And so now they have very complex sort of standards where for high-risk water, you can't have any E. coli for certain uses that have direct contact, the corrective actions that need to be taken. Then there's these other water that it has to have a geometric mean when you've taken multiple different samples below some level. It's super complicated and hard to even follow, even if you know the science, and much less for a lot of growers who are just tell me what I need to do. And then there's also whole rules about how often you test, annual surveys, what the corrective actions are. And I can tell you, we did a whole study, was where do you test, and where do you collect samples, and if you collect from this location versus that location, you're more or less likely to find E. coli in your water. So this is a very imperfect system, basically. But if you get to this sort of the difference in approaches, there's the GAP approach where you're kind of wanting to do an audit. It's all voluntary. You can get a USDA licensed auditor to come do an analysis, audit your good agricultural practices on your farm. Whereas the produce safety rule, it's regulatory, it's mandatory. It's people coming out and coming from the outside. You don't get to hire them. They come in. They're going to examine your farm. But as you can imagine, that's a lot of work to do that and to do it right. And in terms of enforcement, there's just not personnel at FDA to go to every farm and be enforcing the produce safety rule. So a lot of this comes back to growers needing to sort of voluntarily do good practices and pressure each other to do the right thing. For many years, I was following this. I kind of just updated this last night because this was it was like over and over again. They kept saying well, the compliance dates, we're pushing it back. Now, this is the most recent that I downloaded last night. There's this whole thing do you have produce? If you're not having produce, your farm isn't covered by the rule. It goes all the way down. Now it's, does your farm, on average, in the previous 3 years, as defined per section 112.5, have less than 500,000 annual food sales, and a majority of the food is directly to qualified end users, you can be exempt. But if you're more than $500,000 in food sales, then you have to comply, because there's all this concern about how this is going to hurt small growers. So you can see how there was this idea we've got to regulate water quality on the farm. And after many, many years now, it's been changed and adjusted and updated so many times, and now it's kind of like big farm producers that have the capacity to create a very large outbreak are needing to comply with these different rules. And then you go to enforcement. It's okay, well, what's the enforcement? You can have the issuance of advisory letters, but I think it's really, there's now a way to really punish the bad actors. So you can, if there's growers that repetitively have infractions, then that can actually take them to court and have administrative action. So there is now a legal way to deal with bad actors, but it's very much not applied across the board to all growers. So that's kind of where it's at. And that's what I have. Question in the back? Florida didn't, yeah, so Georgia has a high rate of salmonellosis. Florida doesn't show up that way, but they do, yeah. Are there consequences for having that high rate? No, not really. Not specifically. Florida is kind of notorious for not reporting their cases. They don't want to enter the data in the system, partly because they're don't want everyone to know that there's a problem. There's some politics there. But the repercussions are, the repercussions ultimately are if there are outbreaks traced back to food, people won't buy food from that area. So it really can have really big economic damages to the industry. And so far, even though there's all that salmonella in the environment in Georgia and Florida, there haven't been a lot of outbreaks traced back to food production of salmonella. There have been some, but it's not been widespread as it has been in terms of the E. coli in these areas that are producing leafy greens is a much bigger public health problem. So I don't know if that really answers your question, but.